Good morning, and to our East Coast friends, good afternoon. Welcome to ERA's webinar, Keys to Improved PT Results on Your Wastewater Microbiology Samples. Thank you so much for taking the time to join us today. My name is Tanya Ron, and I am an account representative here at ERA. Before I turn over the webinar to Mike Blades, I have just a few housekeeping details. First, all attendees are in listen mode. So if you have a question during Mike's presentation, please type your question in the questions box. We will gather and answer as many questions as possible as time allows at the end of the webinar. Also, the webinar is being recorded and will be available for viewing on the ERA website. You will receive a link once the webinar recording is posted. So, a little information about ERA. ERA was established in 1977. We're headquartered in Golden, Colorado, just 10 miles west of Denver. Our facility here in Golden includes more than 25,000 square feet of dedicated laboratory space with areas dedicated to inorganics, organics, and customs. ERA is a provider of proficiency testing products and certified reference materials to more than 7,000 environmental laboratories in more than 80 countries. Our products are available in a variety of matrices, including wastewater, drinking water, air and emissions, soils, and more. ERA is a T&I accredited PT provider. We are also a certified reference material producer and a chemical testing laboratory by A2LA. And we are registered to ISO 9001 by the National Quality Assurance Organization. In addition to serving environmental laboratories, we also manufacture TOC and conductivity reference materials and calibration standards for pharmaceutical, medical device, and biotech manufacturers. I want to start by giving a very brief explanation of two key terms that can be very confusing: PT, or proficiency testing, and CRM, or certified reference material. Proficiency testing samples are blind standards designed for use for laboratory accreditation and certification. These are the samples that you will report back to ERA. Certified reference materials, also known as QCs or quality control standards, are standards of a known value, used as a check on your internal quality control processes. These are the samples that do not need to be reported back to ERA. CRMs or QCs can also be used for demonstrating analyst capability or used to troubleshoot instruments and or method issues. During today's presentation, Mike will give you a very brief overview of a few key points on improving your PT results. And as I mentioned, we will answer questions as time allows. Now, time to turn over the presentation to Mike Blades. Mike is ERA's Organics Product Line Manager, and he's responsible for our Organics product line, which includes microbiology, radiochemistry, and all organic products. As a graduate of the University of Michigan with a BS in biology, he's worked as a chemist for more than 26 years, and he's been with ERA for more than 19 years. And now I would like to introduce Mike Blades.
Thanks, Tanya. Good afternoon, everyone, and welcome to ERA's webinar on getting better PT results for wastewater microsamples. Today on the agenda, we're going to cover four main areas. The first three we'll go over the NELAC criteria for wastewater microsamples. We'll go over calculating PT limits using the NELAC criteria, and we'll go over ERA's sample design and our intended use. These three areas are really just to give you a general understanding of why do we make our product the way that we do, how are they evaluated, and what is ERA's product. This is really just to give you a good foundation to make sure that everyone understands, has a basic understanding, so that when we do move on to our main focus, which is really keys to improved results, everyone will have a similar background. The other final topic that we're going to cover is questions. Hopefully our presentation is going to cover most of the issues, but at the end we'll try to get to as many of the questions that you have as time will allow. All right, the NELAC criteria. The National Environmental Laboratory Accreditation Conference, sometimes referred to as the NELAC Institute, they define the required target analytes, manufacturing ranges, acceptance criteria, and PTRLs that are used by all approved PT providers to design their samples and evaluate the results. These are all communicated to the PT providers via what we refer to as fields of proficiency testing tables. The one on this particular screen is for the non-potable water table, which covers primarily wastewater. These are posted on TNI's website. They're available for everyone to look at. The origin of these tables, they've been put together by committees that were formed by TNI. They were groups of experts representing the EPA, state agencies, the PT providers, the laboratory community. They got together and actually defined this, not just for microbiology, but for chemistry and all the parameters. And this is really the skeleton of the criteria that drive the proficiency testing samples. The table that you're looking at right now currently defines 4 analytes. You'll see total coliform, fecal coliform, E. coli, and enterococci. You'll see that they're listed by 2 separate technologies. MF is membrane filtration, MPN most probable number. This gives us 8 separate analytes that the PT providers must treat individually. When I say that, what I mean is that we must collect the data separately and evaluate them independently from each other. The primary reason that they split these analytes apart was because primarily different methods produce different results with different standard deviations. So in order to treat everyone fairly, they're split apart. Everyone isn't lumped together. So currently, those are the 8 analytes that are defined, and we need to treat all of those independently. So to add a little bit more clarification to that, if you were running method 9222B for total coliforms by membrane filtration, you'd be reporting the result as total coliform MF. If you were doing 9222D, fecal coliform by membrane filtration, it would be reported as fecal coliform MF. So that should kind of give you the idea of how things, 9221B would be an example of total coliforms by multiple tube fermentation. That would be reported as total coliforms MPN. So all of these data points that we collect need to be sorted according to how they're being reported. Now also on this table you have a design range that's given for each of the analytes. This is a range that the PT providers will target for each of the lots that they manufactured. It's required for us to vary it from study to study over the studies that we produce in a given year. So you can see that the range is 20 to 2400. In any given one study, we may be designing the PT sample to fall towards the low end of that range, the mid, or the high. It's a requirement that our accreditors will look at to make sure that in fact we're doing that. We can't always make it at the same level. So just depending on what study you're enrolled in, you may find something that's lower or higher. It's something that's varied. Also on this design range, it's important to keep in mind that it is in fact just a range that we're designing our product to. It's not a range of acceptable results. A study designed at the lower end of the range may have a range of acceptable results which actually go outside of that design range. So if we make something at 20, it's very possible that the range of acceptable values will be below that. Or conversely, if we made it on the very high end, it's possible that the range of acceptable results would go above the high end of the range. So it's important to keep that clear. The other thing that you see presented on this table is the PTRL. It's presented as the number 2 CFU or MPN per 100 mils. This is given to us by NELAC basically as a guide for the laboratory for PT samples. It gives you a lower level, well, I should say, it's the level at the lowest acceptable result could be obtained from the lowest spike level. So if we were to manufacture a given lot at the very low end of the range, you should not expect to see an acceptance range that would go below 2. So it's never going to go down to 0 or 1. The other area that's covered on that table is the acceptance criteria. It's listed as the log transformed plus or minus 3 standard deviations. That's the acceptance criteria that all the PT providers have to follow when it comes to non-potable microsamples for all of those 8 analytes. Let me try to explain that. Essentially what that means is that all the data reported by the participants needs to first be log transformed. Once you've log transformed the data, you'll calculate the mean, the standard deviation, and then you'll calculate an acceptance interval, plus or minus 3 standard deviations. Then you'll evaluate all of those results, and then they all get converted back to the whole numbers before the reports are sent out to the customers. Now you may be asking yourself, why would they do that? I've included a graph on this slide to try to illustrate why you want to log transform microdata. Essentially, this graph is a frequency plot. You can see 2 lines in there. One's labeled as non-log data. The other is labeled as log transformed data. Generally, when you deal with chemical analyses, the distribution of analytical results that you get follow a Gaussian curve, or what people refer to as a symmetrical distribution of values about the mean, kind of the bell curve as you would. The non-log transformed data, you can see it's not bell-shaped whatsoever. Generally, microdata is not symmetrical. It's very rarely that you do get a symmetrical data set. The bacterial counts often are characterized as having what they call a skewed distribution. You get a large number of low values and a few high ones. And really, this is the nature of the test methods when you're sampling a biological population. It has a lot to do with the selectivity and differential nature of the tests themselves. So what you get in a data set like that is you get an arithmetic mean that's considerably larger than the median. So if you're going to evaluate this data set, in order to treat everyone fairly, you need to somewhat normalize the data set before you apply the plus or minus 3 standard deviation limit. The non-log transformed data, you can see, starts to take a much more bell-shaped curve. So again, all data is treated exactly the same way, but you get to treat both the high and the low data points that are resulted more fairly. It's critical that you do this. If you don't do that, you're really going to discriminate against certain data points. So it's important to keep in mind that all the PT providers are evaluating the data by the NELAC criteria that were given, and this is specifically what needs to be done with the data. Okay, now I wanted to talk a little bit about the sample design and the intended use. ERAs, QCs, and PTs are delivered to our customers as lyophilized pellets in glass vials. They're accompanied with a phosphate rehydration fluid. ERA has been supplying their product in this particular format for about 13 years now. Each of the vials will also contain a desiccant pouch that will aid in the preservation of the sample by reducing any kind of possibility of moisture. For the non-potable water, ERA has their colorform sample, which is designed in the 20 to 2400 CFU or MPM per 100 mil range. They also have an enterococci that is designed within the 20 to 1000 CFU or MPM per 100 mil range. Remember that these are the design ranges, they're not the range of acceptable results. ERA's quantitative products have been validated to be unaffected by ambient temperature shipping conditions up to 7 consecutive days. ERA's normal shipping service is the second-day service, but the samples are stable well beyond that normal shipping service. So we routinely ship things ground. We ship these all over the world these days. So having a sample that is designed in a way that allows it to be very stable at ambient temperatures is really important in order to supply all the participants a equivalent challenge. Now, once you receive the sample, the pellet does need long-term storage at 4 degrees C. That's just to ensure the integrity of the sample until you get ready to analyze it. As a PT, that may be analyzing it on the 44th day of a 45-day long study, or if it's QC, it might be 6, 8, 9 months after you receive it. It's really, you're putting it in the fridge to give it long-term stability. The phosphate buffer that comes along with all the samples is your typical phosphate buffer with mag chloride. It doesn't require any refrigeration. You can just store it at room temperature. That's how the instructions read. People have put it in a fridge. It's not going to cause any problem, but really there's no reason to have it there. And when you get ready to use it, you have to warm it up to room temperature anyway. So please just leave it out at room temperature. Now, the expiration date on ERA's product is 3 years from the actual manufacture date of the pellet. Obviously, that indicates that it's a very stable, very robust product. The reason we lyophilize our samples is because it is one of the truly recognized techniques that ensures the integrity of the organism over long periods of time. Doing it in any kind of modified process, whether that's a controlled dried process like a vitroid or a lenticule, or it's a whole volume, all of those are, do involve some preservation, but they're less recognized as really ensuring the long-term integrity of not just the viability of the organism, but the integrity. anything shy of true lyophilization kind of opens you up to the possibility of the organisms actually going through some changes that you want to avoid, definitely for a QC and a PT sample. I also wanted to mention that the organisms that are used in ERA's wastewater product, just so that everybody was aware, the total fecal and E. coli analytes are accomplished with ERA using E. coli as our target organism in those samples. The specific culture collection that we use for that sample design is NCTC 11954. We've been using that for many years. For those of you that are not familiar, NCTC is the National Collection of Type Cultures. It's a culture collection out of the United Kingdom. It is been around for a very long time. It's very well recognized. For the Enterococci sample, we use Enterococcus faecalis, NCTC 775. All of that information comes on the certificates that we supply with the product. It also is disclosed on any proficiency testing report that you get. The product that we're providing, it's intended to be used as a proficiency testing sample or a quality control check of the entire analytical process. It's not necessarily intended to be used to start stock cultures, to rehydrate directly onto media. It's really designed to create a sample and challenge your overall method. So that's how we intend it to be used. I also wanted to give you kind of a general overview of the instructions on how we ask you to use our product. It's quite simple to use. Basically, all we ask you to do is you remove the pellet from the fridge that you've been storing at 4 degrees C, warm it to room temperature. It generally will take about 30 minutes to do that, assuming that you have a fairly comfortable laboratory. Once you've warmed that to room temperature, you should already have your buffer solutions already at room temperature. So all you need to do is aseptically transfer the pellet from its glass vial over to the buffer. You can accomplish this in a variety of ways. I've been doing this for years and I'm pretty comfortable with. I just pour it, and it is quite simple to do. You could use a pair of sterile tweezers. I've heard of a variety of techniques over the years, but you basically want to make sure that you don't contaminate the sample when you transfer it. At that point, the pellet will almost dissolve upon contact with the surface of the buffer. Close the top, shake for a few minutes. Generally, 10 to 20 seconds is more than sufficient, and it's ready to perform your analysis. Okay, now we want to kind of get into the meat of it. Hopefully those explanations of our products and the evaluation criteria have given you just a basic background of how our products are used and intended to be used. Now I wanted to actually get into some of the areas that we've seen over the years that if people don't have in place, they can have trouble. So I wanted to start out first with just some general quality control criteria. And the first thing I wanted to start with is just a general disclaimer. I hate to do this, but I want everyone to be aware that the issues that I'm going to cover are intended to get you to think about what's important in microanalysis. I'm not bringing them up, nor do I intend them to supersede any of your current procedures that you have in place. My real intent is to make sure that you're thinking about some of the issues that we've seen come up with people that are getting poor results. So the first thing that I wanted to cover is sterility checks. Laboratories should perform sterility checks for the procedures that they have in use. Kind of goes without saying, but I need to say it. For membrane filtration procedures, you should check the sterility of your media. You should check membrane filters. You should check any buffered dilution water that you're using. If you're using pipettes, flasks, petri dishes, basically all the equipment involved in your process, you should be checking. Generally, it would be good practice when you're doing the analytical procedure, depending on which procedure you're doing, you may want to check for sterility at both the beginning and the end of a series. You could do this with using buffered dilution water as the sample. An example would be if you were doing a membrane filtration and you only had 1 or 2 filter flasks and you were going to use those filter flasks throughout the process with just rinsing in between. You want to make sure that that's an effective technique. So you incorporate some sterility checks to make sure that you're not getting any carryover from one sample to the next. For multiple tube procedures, again, you want to check sterility of media, buffer dilution, your glassware. Each new batch of any of those materials, when they show up, you should check before you put them into use. The next area would be positive controls. For each new batch of media and method apparatus, you should check the analytical procedure by testing a positive control to demonstrate that the media produces the expected reaction to the organisms under test. You really want to ensure identity, traceability, reference cultures that you're using. You need to make sure that they're coming from some recognized traceable source. There are folks that will use in-house stock cultures that they are maintaining. There's limits to that. You want to make sure that you're not passing that too many times. You can have issues that will come up. So if you can't start with something that is ideally fresh each and every time and perfectly identifiable, traceable reference culture, you just want to remember that that is an issue. You should always be challenging media and new equipment to make sure that you don't have an issue. They could be, the cultures could be like a single-use preparation, or again, cultures that you maintain. You just want to make sure that, you know, you have a good purity and good viability of the organism. ERA sells some QC samples that are intended for that purpose. There are a lot of other companies out there that do as well. The important thing for you to remember is you need to be checking all of these things before you move forward. If you're assuming that your media is just as good as what it was the last time, relying solely on the manufacturer, you may be making a mistake. A little bit on positive controls, not to backtrack, but I'm going to backtrack. Standard Methods has a variety of cultures that they recommend. You may want to look at those. I've mentioned to you the strains that ERA uses for their PTs and QCs. That may be what you want to use. Your state agency may tell you what you should use. You may be using some of the commercially available methods out there. If you are, you may want to follow that manufacturer's recommendations. Okay, on negative controls. Each new batch of media, the method apparatus, you need to check the analytical procedure by testing a negative control to demonstrate that the media does not demonstrate the typical positive reaction for that target organism. So if you're expecting it to produce an atypical colony, it should produce an atypical colony. Your negative control should be an organism. It should be something that, you know, either recommended by Standard Methods or recommended by a standard manufacturer, but generally it would be something that you would get some kind of reaction with your media, either turbidity with no gas production, an atypical colony, something along that line. Some of the negative cultures that ERA uses in some of their products are Pseudomonas species. Generally fairly good for most of the various wastewater methods that are out there. But again, you want to use something that's appropriate for the method that you're using. And again, it could be a single-use preparation, or it could be something that you maintain a stock culture. But the key is that you need to maintain the purity and the viability of it. You don't want that organism changing over time and giving you a false reading of some sort. Okay. Method evaluation, or method evaluation or validation. Does the procedure produce the expected quantitative result? All of the challenges that you're getting in non-potable water are quantitative challenges. So when you do evaluations, when you first put a method into place, you should be challenging it quantitatively. You want to make sure that it's giving you a result that would pass if it were a proficiency testing sample. You can do that by either buying a quality control sample, or if you're using a stock culture, you could come up with a dilution scheme that would ultimately dilute down into a water sample that would challenge it at an appropriate level. So an appropriate level for wastewater type testing, at least when it comes to the PTs, is 20 to 2400. If you're challenging your method with basically a sample that is in the 10 to the 7th range, you're not really evaluating it at the level that would be appropriate for passing a PT. You may have issues with your method or your media that you would see a too numerous to count plate, but if you were to dilute that down into, say, 100 counts, you may see that the media may not be up to snuff. So method evaluation is really important, and you want to challenge it appropriately. Hydration of the pellet. I wanted to touch on this to make sure that everyone understands that you should follow the instructions that you're provided for PTs and QC samples. The reason you don't want to deviate from that is because, as we've discussed with the evaluation criteria, when it comes to a PT, you're evaluated based on the mean of the study data plus or minus 3 standard deviations. The assumption is that everyone is following the instructions in the same fashion. If you decide to do it in a way that is completely different from your peers and you're a participant in that study, it's possible what you do could produce a result that is different just because you were off on your own doing something different. So it's really important to follow the instructions that you're given. Our sample comes to you, as I've discussed, lyophilized with a phosphate buffer. Once you rehydrate it, you've got about 30 minutes to perform your analysis. The reason I say that is because the pellet does have certain carbon sources in it. And when you dissolve that in the phosphate buffer, there is the possibility that the organisms, once they are somewhat resuscitated, could use the source of carbon in the pellet as something that will cause them to grow. So you definitely have to be somewhere beyond 30 minutes before that becomes a concern, but it is potentially a concern. Don't rehydrate your sample and then go out to lunch and then come back and cross your fingers. It's an area that you need to follow the instructions. Okay. Culture media. The quality of the media when it comes to getting the right answer on the sample is probably one of the most critical aspects of getting the PT or QC to work out. You should never prepare your media from raw ingredients if the source of dehydrated media is available. If you're preparing it from dehydrated media, follow the instructions closely. Critical aspect, if you're told to check the pH, you really need to check the pH and make any adjustments. Organisms are very finicky when it comes to pH. You want to make sure that you're checking it. You always want to evaluate the media before you put it into first use. You never want to use media outside of its expiration date. You never want to use media that hasn't been stored according to the manufacturer's specifications. If they store it out of the light at ambient temperature, you need to do that. If they say store it in a fridge, you need to do that. These are the source of carbon that you're going to grow your organisms from. If you do anything that starts to break down this media, you're going to have trouble. So make sure that you follow the instructions from the manufacturer, both when it comes to manufacturing it and storing it. Proportions. Make absolutely sure that if you're supposed to weigh out a certain number of grams to a certain volume, follow it very, very closely. If you start deviating from it, you're basically creating conditions that are going to be different from you than they are from other participants in the study or the folks that analyze the QC sample. Okay, dilutions in rinse water. As I'd mentioned a little bit earlier, pH conditions are critical. You only want to rehydrate the pellet that we provide you with the buffer that we provide you. All of our quality control, all the other participants, everybody is using that same buffer. You want to make sure that you use it as well. If you're doing any kind of dilutions or rinsing of your apparatus, you don't want to use just deionized water or distilled water. ERA, standard methods, general good micro practice is going to recommend that you're using phosphate buffer or peptone water. The reason is it's not uncommon for DI or distilled to the pH to be, well, something other than what would be ideal. And that can cause inhibitory effects on the organism. Take a look at standard methods, section 9050C. It'll give you the instructions on how to make buffered water or peptone water. The other general quality control criteria that I wanted to mention was thermometers. Temperature is critical. You want to make sure that you're using thermometers that are calibrated and are incremented that are appropriate for whatever method you happen to be using. You want to check the accuracy semi-annually against a NIST thermometer. You want to use any correction factors, if there are any, related to the thermometer you're checking it against. Water bath incubators. You want to verify that the water bath is at the desired test temperature. If it says it's supposed to be within plus or minus 2 degrees C, it absolutely needs to be within that tolerance. You probably want to record it, monitor it at least twice daily, generally 4 hours apart. You want to make sure that it's stable and uniform. Water baths, you want to use ones that have a gabled cover. You want to use circulators. You want to place the incubator in an area of the room where the temperature is relatively stable. You don't want to put it next to the air conditioning vent, or you don't want to put it by a window. Those are all things that are going to cause you havoc when you're trying to maintain such a tight temperature tolerance. The air incubators, again, you want to make sure that you're staying within the tolerance. You want to make sure that samples, cold samples, are incubated at the test temperature for the required time. You want to record temperature on the shelves, the various shelves, if it's a fairly large incubator. It's very possible that the temperature at the top is not the same as the temperature at the bottom. You want to make sure that you know that it's at the right temperature for where you're incubating the samples. Opening and closing the door of incubators. Incubators, depending on what size they are, you're going to have a refresh time. When you open the door, the temperature is going to drop, and it's going to take a certain amount of time before it ever gets back to the incubation temperature. You want to have an idea of what that time is. so that you can kind of compensate your incubation time based on the period of time where it's not actually at the incubation temperature. So get an idea and avoid opening up the door as much as possible. All right, keys to improved results. Want to get into some method-specific issues. Membrane filtration methods. Okay, one of the most important things you need to know about membrane filtration PT results is PT results that are reported as too numerous to count will be evaluated as no evaluation. It's therefore critical that you run the appropriate dilutions. We'd also recommend, if you're running QC, that you routinely run them at various dilutions kind of to simulate the PT scenario. Proper dilution scheme. When you're performing membrane filtration, basically the sample size will be governed by the expected bacterial density. The assigned value for all ERA, QC, and PT lots will fall within the range of 20 to 2400 CFU or MPN per 100 mils. Given that range, you want to select sample portions that will most likely yield a membrane filter count in the ideal range. So depending whether it's a total method or a fecal method, that may be 20 to 80 or 20 to 60. What we would recommend is that you run the entire volume of the sample in portions to cover that range. And of course, the sum volume of all of those should be equal to the total volume of the sample that's provided. That should generally give you a result on one of those plates that falls within your ideal count range, and then you just need to make the adjustment based on which plate you selected. If in the event it's a lot that's manufactured in the lower portion of the range, it may be possible that none of the plates fall within the ideal counting range. If you look at standard methods, you'll see a section that will actually guide you through what do you do in that instance. And basically it just involves looking at all of the plates that you ran to come up with what the number is that you're going to report. A general example that we give folks when they're running this sample, you could run a 50 mil, a 30 mil, a 12 mil, a 5 mil, a 2 mil, a 1 mil. If you're going to dilute those volumes with an additional volume of water, you want to make sure that it's also buffered water. Just if you're doing a filtration test, you don't want to pull too low of a volume through your filters. Most of your procedures probably will give you guidance as to what that volume can go down to before you need to add additional diluent. Okay, filter paper. Generally, folks are using 47 millimeter diameter filter paper, 0.45 micron pore size. Filter paper can be a real issue for pore recoveries. Generally, when you've got an issue with your filter paper, you're going to see the colony development be out of the ordinary. You may have a colony that'll go to a grid line if it's grid-lined paper, and it changes directions. That's a good indication that there's something wrong with your filter paper. You may get abnormal spreading of the colonies. You may get poor or absent colony sheen. Keep in mind that I've told you the organism that we use every time for the wastewater sample. You should never expect to see any kind of atypical results. These samples are not tricky in that way. You're always getting the same organism every time. So if you don't get good sheen, sometimes it can have something to do with the filter paper itself. You may see brittleness in the paper. When you do your initial evaluation, you may see decreased recoveries in what you normally see for that quantitative challenge that I talked about earlier. You may get wrinkling on the paper. You may get areas of the paper where you get absolutely no absorption of your media. If you don't get any absorption of the media, that means that any organism that's seated on the filter in that area does not have any food to grow. That obviously will affect your results. It's not going to develop a colony that's going to be countable. So if you get kind of a tie-dyed look to your filter paper, it's very possible that you got a bad batch. Another little trick that's in standard methods, they talk about taking a filter and floating it on reagent water. You can take a Petri dish, put in a small amount of DI water, take a pair of tweezers, float the filter on top of the water. You should see it uniformly diffuse in about 15 seconds. If your filter floats like a little boat there for a minute, you got something wrong with your filter paper. It won't absorb the nutrients in a way that are going to get the organism to grow. Media broth for broth and pad. Pads must retain between 1.8 and 2.2 mils of broth. That's defined in standard methods. You need to confirm that if you're getting media that's pre-mixed in vials and you're cracking the vials and dumping them out. Make sure that what you're delivering to the pad falls within that tolerance. You can do that by just measuring how much are in the ampules to begin with, pour it onto the filter. If you have broth that doesn't get absorbed in the filter, measure how much of that, some people call it pour off, still exists. If it puts you outside of that tolerance, it's possible that those pads have become compressed over time or have absorbed moisture. If that happens, you're going to make it difficult for you to get a good recovery because you're not delivering the amount of nutrients to the organism that really is going to be ideal for growth. Application of filter. It's really important that you get good contact between the filter where your organisms are seated and the pad that has the absorbed nutrients. If you don't make a good contact between those two, there's no nutrients for your organisms. So you need to put it on very carefully. Generally, what's recommended is to get maximum contact as you roll it from one edge to the other. So you take one edge, you put it on the pad after you put your media on, and you slowly roll it onto the pad, making sure that you don't trap any air underneath it. Once you've completed rolling it on, a good practice is to tap the Petri dish on the lab bench. That'll add an additional kind of contact between the two. If you don't have a good contact between your filter and the pad, your organisms will not grow. They don't have the nutrients. They're sitting up there with no lunch, so to speak. Okay. Keys to improved results related to multiple tube methods. I wanted to get into a few issues here. Again, I also wanted to mention if you're doing a tube method that involves a pre-enrichment with some type of membrane filtration technique, refer to what I was talking about with the membrane filtration techniques related to that portion of it. This is primarily focused on doing tubes. One of the most important things that people need to know when it comes to multiple tube fermentation is a greater than or equal to value will be scored as that value. So if you're doing a method that say 15 tubes, where your maximum value, if you get 5 positives in each of your 3 series of tubes, is 1600. If you report greater than or equal to 1600, it'll be scored as 1600. If you recall, the manufacturing range that PT providers are required to use is 20 to 2400. You're basically using a method that doesn't cover the entire manufacturing range. So if you're analyzing a PT that happens to be in the higher portion of the range, you may have cut yourself short. You basically aren't covering the entire range. Now, Dilution scheme. If you're performing multiple tube fermentation, you want to run a series of results that's consistent with that range. So as I mentioned earlier with the 15 tube, standard methods does allow you to run an additional series of tubes to increase the range that you can cover. So you may want to consider that given how the PT samples are designed. You can basically, instead of just running 10, 1, 0.1, you can do an additional 0.01, and standard methods will give you guidance on how you shift the interpretation of your positive tubes, and then you go to the MPN table and you adjust that value accordingly. So that may be something that people doing the old 9221 method may want to consider when they're running unknowns, PTs. Another issue that comes up periodically is degassing of broth. To avoid false positive results due to dissolved gas in the media, you want to make sure that you're at a minimum pre-warming your tubes up to room temperature, and then if you do get any outgassing of the media, you want to get that out of your Durham tube before you actually do your inoculation, or out of your fermentation tubes. You don't want to have you don't want to inoculate the sample, put it in, say, a water bath, have it warm up the media, have oxygen come out of the media, and you end up with this little bubble in your tube, and you interpret that as a positive result. You want to make sure that you've eliminated that possibility. So degas, the other thing you may want to consider is before you inoculate samples, you get the tubes to your incubation temperature. That will avoid that significantly. The other issue I wanted to mention has to do with broth concentration. If you're doing an MPN method, you want to make sure that you're doing excuse me, 1 second. Sorry about that. I was losing my voice. So broth concentration, you want to make sure that you're making it at the appropriate strength for the method that you're using. If it's double strength that's required, you want to make sure that you're doing it. If you're not doing it at the right strength, it's very possible that the aliquot of sample that's being used, you can end up with a negative result just because essentially you're not following the method. Okay, keys to improve results specifically related to enzyme substrate methods. A lot of you are probably familiar with Quantitre. This is an area that I want to try to address some of those issues. Okay, the first area, when you're dealing with most enzyme substrate methods, there are a ton out there: ColAlert, ColAssure, ColAtag, ReadyCult, Ecolite. There are a variety. They have interpretation time windows when the results are considered definitive. An example would be, I believe ColAlert 18 uses an 18 to 22 hour window. ColAlert uses a 24 to 28 hour window. Basically what this is saying to you is that ColAlert 18 is not really an 18-hour test. At the end of 18 hours, if the result has changed, it's yellow or it fluoresces, that's a definitive result. If it is not, then it means that the result is still could potentially be definitive out through whatever that read time is. So even though you get these products and your standard practice may be, well, we always read them at 18, you may be cutting yourself short because technically the manufacturer still considers the result to be a positive result if it changes within a certain time window. It's not until you get outside of that window that a negative result is considered definitively negative. So make sure that you're aware of that. Another issue when you're dealing with QuantiTrade, there's a couple of different formats out there. One is a 51 well, the other is a 97 well. You want to make sure that the range that's covered by the number of wells for the format that you're using covers the manufacturing range. Now, if you use a 51 well, you're not actually covering the entire manufacturing range. It has an upper capacity. The 97 well actually has a capacity that'll cover the entire range. Doesn't necessarily prevent you from using the different formats. You just need to be aware that you're not completely covering the range if you run the sample at 100%. Comparators. Ideally, every manufacturer of enzyme substrate method would be required to have a comparator. Fortunately, they're not, so not all of them do. If there is a comparator, you need to use it. As most of you know, a comparator really distinguishes the threshold for what they consider to be a positive result from a negative result. You should use them if they're available. If you don't have them available, what I would recommend is that you use a quality control sample to run along with it, and you use that as a point of reference as to what constitutes the chromogenic or fluorogenic change. Another issue that I wanted to cover related to enzyme substrate methods has to do with QuantiTrade, and they now have what they refer to as a fecal addendum, where you can use ColAlert 18 in combination with a QuantiTrade and incubate it at a 44.5 degrees C for the determination of fecal coliforms. Normally that test incubated at 35 degrees C determines total coliforms and E. coli. By incubating it at the elevated temperature, it's been written in the method and not completely approved, but approved some places, that you can determine fecal in that fashion. One thing that we've seen is that you need to be really careful when you're doing this because it's very sensitive to temperature. You want to make sure that, in our opinion, you're ideally using a water bath, but definitely maintaining the temperature tolerance prescribed in the method of plus or minus 0.2. If you get outside of that, it's very possible that you can cook the organisms and they will not grow. One way to help you to avoid from cooking the bugs is, if you're using a water bath, use a circulating bath. Keep the trays off the bottom or off the walls. Make sure that the trays are incubated facing up. And remember that you're looking for a color change only. You're not looking for fluorescence. These things will not fluoresce under those conditions. The reason we tell you to incubate them facing up is because the wells can trap water, and that water can get hot and it can cook your bugs. Okay, now as I promised, I would like to open it up to questions. And hopefully I didn't go too long, so we'll try to get to these, many of these as possible. All right. Our first question: I have difficulty correctly identifying fluorescing wells using IDEX SimPlate for HPC method. What do you recommend for troubleshooting? Generally, the HPCs may be foreign to most of the wastewater folks because usually it's a drinking water parameter, but there are some folks out there that do have to do some HPC methods. We do occasionally make it available for some of our participants in some of our studies. So I've done the method. It can be really tricky. So those of you that don't know, IDEX SimPlate, you're looking for a chromogenic, or actually a fluorogenic endpoint. and there is no comparator available for it. So as what I kind of mentioned in the presentation, I would recommend that you run a QC sample along with it. Another thing that will help you out is IDEX sells a viewing box. We call it a dark box. Basically, you put the fluorescence lamp on the top of the box, and then you look in through a viewing port, and you hold the sample inside the box. It makes it a little bit easier to read SimPlate results. The other thing that I've got in the habit of doing is I read it from the reverse side of the plate. It's easier for me to do, and that way I can check the wells as I go. The other thing, I guess actually probably the best thing, is you probably run a QC sample to use as a reference. The other thing, you may want to call IDEX and tell them they need to come up with a comparator, because that really would be important. Okay, I have another question here. Do you have suggestions on how to have a better count on fecal coliform MPN? Well, hopefully I've covered a lot of that in the presentation, and I may have mentioned this. Probably the most significant thing that I've seen for people doing fecal MPN, if what we're talking about here is a 15-tube test, is to run that additional series of tubes. It's generally going to give you a more accurate result. When you're only doing the 15 tubes, you're limiting the range that you are covering, and also by doing that additional set of tubes, you add an additional level of accuracy to that test that you don't normally have. So that would be what I would recommend. Okay, I'm being told that we're out of time. I want to thank everyone for joining us today and submitting their questions. I wish we had more time to answer all of them. As Tanya had mentioned earlier in the webinar, a recording of the webinar will be posted on ERA's website by the end of the week. We'll receive an email. You'll receive an email with a link when it is recorded and available for viewing. We'll also post answers to the questions that we did not have an opportunity to address. In the meantime, you can contact ERA by calling 1-800-372-0122 or by sending an email to info@eraqc.com. Again, thank you for joining us, and this concludes our presentation.